Hi everyone, I am Ömercan Ünüvar from Bolu Abantiziyet Baysal University, Department of Chemistry. I'm going to present our research titled as Novel Bacterial Strains Associated with Wheat Roots. I thank to the committee of the International Eurasian Conference on Biotechnology and Biochemistry. I am going to give some information about wheat and fertilization before giving our aim, methods, and the results. As you know, wheat was first cultivated more than 10,000 years ago and Fertile Crescent. You can see the Fertile Crescent in the map. Today, it has provided fundamental food to more than one-third of the world's population. Wheat is common and important because ease of handling, storage, and processing, and due to the wide adaptation capabilities. Wheat is the basic food in the developed and especially in developing countries today. You can see in the slide some general information about einkorn wheat. The data from archaeological studies represents that wheat firstly occurred in the southeast region of Turkey, Lebanon, Syria, the Levant, Israel, Egypt, and Ethiopia. Einkorn, the first cultured wheat, domestication dates back to 10,000 before the Christ in Turkey, which is earlier than the existence of wild barley. During the Bronze Age, its cultivation decreased and nowadays rarely planted. It is found in the market as a half food which reduce and prevent chronic diseases such as diabetes, cancer, Alzheimer's, and cardiovascular diseases. After that part, fertilization, firstly chemical fertilizers and secondly biofertilizers. Fertilization plays an important role in the production of wheat, as in many agricultural products. The survival of plants depends on the presence of sufficient nutrients in the soil naturally. However, the amounts of mineral substances are not always sufficient. Especially in the soils grown on the plant, these nutrients decrease with time. For this reason, the nutrient elements that are decreased in the soil must be enhanced to be able to get more quality from the cultivated plants. Plants take significant amounts of nutrients from the soil. General damage caused by the intensive and unconscious use of chemical fertilizers can be listed as pollution of ground and surface waters, negative effects on the physical, chemical, and biological properties of soil, negative effects on indirect or direct plant growth, nitrate accumulation in foods, and global warming. Because of the negative effects of the chemical fertilizers to human health, biodiversity, and nature, the agricultural processes in which use biofertilizers are developed. The biofertilizer can be defined as a substance which contains microorganisms which when applied to seed, plant, or soil, colonizes the rhizosphere or the interior of the plant and promotes growth by enhancing the supply or availability of primary nutrients, such as nitrogen, phosphorus, etc., to the host plant. Pseudomonas, Bacillus, and Burkholderia species are important because of plant growth promoting effects and also antagonistic effects against plant pathogens. The major challenge for using biofertilizers in agriculture arises from the growth incompetency of isolated bacteria in various environments. In this study, we hypothesized that growth of einkorn wheat can be promoted by using the bacteria isolated from rhizosphere of local wheat species, locally called ESA wheat. The aim of this study is isolation, purification, and identification of the natural bacterial strains from the rhizosphere of the local wheat grown in Seven, Bolu. For reach to our aim, firstly bacterial species were isolated from einkorn roots by using phosphate buffered saline 20 and sonication. You can see our procedure in this slide. Then LB agar media was used to differentiate of the bacterial colonies until obtaining pure colonies in a plate. You can see in this slide the procedure of the LB agar preparation. Identification of the microorganisms were studied by using three different approaches. multi-tough identification in Industrial and Technological Application and Research Center of Bolu BioTechBioChem University, and 16S ribosomal DNA amplification, and 16S 23S ribosomal DNA intergenic region amplifications. You can see the universal primers for these amplifications in this slide. These are our PCR reaction mix and condition. Most of the researchers apply one type of identification method. On the other hand, in this study, triple verification was used to identify the putative bacterial species by using multi-tough results and NCBI blast results. According to the results, our first bacteria identified as only its genus, Artobacter, and you can see our lab code with the genus in the slide. Second genus name is Artobacter again. And our third putative bacteria is Pseudomonas. Fourth identification result is Pseudomonas. Fifth result shows that its species name is Pseudomonas poae. Next is Pseudomonas fiervalensis. After that, again Pseudomonas species. And after that, the next one is Bacillus subtilis. Next one is Microbacterium foliorum. And the final identified genus is Microbacterium. Let me briefly summarize our study along with the results, step by step. First is sampling, and then the isolation procedure, and then the identification procedures. And you can see the results on the right side of the slide. In the study, we were able to confirm species name for 10 bacteria studied. On the other hand, each approach predicted species name for 7 bacterial species. The names did not match among different approaches. Thus, we prefer to name them with their genus name and stock IDs of our laboratory in Bolu BioTechBioChem University. In the future, the efficiencies of 10 bacteria for their potential to promote plant growth, nitrogen fixation, phosphate solubilizing capacities, and indole acetic acid formation are going to be investigated. Also, whole genome sequencing analyses are going to be carried out to define exact species names and elucidate the biotechnologically valuable gene products. This project is supported by Oz Scientific Research Company established in Bolu Teknokent, which is funded by TEDEP TÜBİTAK, with project ID number 2180200. Thanks for listening to me, and healthy days for you and your loved ones. Thank you.